mutations of the tumor-suppressing gene BRCA1 lead to an increased risk for breast cancer, as well as melanoma, prostate, and pancreatic cancer. Juhas and fellow researchers proposed a method of inhibiting DNA polymerase beta to determine if it was synthetically lethal in BRCA1 deficient cells. They utilized siRNA and a biacetate pro-inhibitor to knock down the expression of DNA polymerase beta. Once they confirmed it was a synthetically lethal partner with BRCA1, they compared the efficacy of the pro-inhibitor with olaparib, which is a drug that inhibits poly-ADP ribose polymerase and is already approved by the FDA. What they found was that the suppression of DNA polymerase beta using the pro-inhibitor was more effective in killing cells missing the BRCA1 gene and was less cytotoxic in cells that present BRCA1 as compared to olaparib.
Bisphenol A, or BPA, is a common compound used for epoxies in plastics, but has been shown to be an endocrine disruptor. Hambleton and Stanzione synthesized a lignin-inspired alternative to BPA they named bisguaiacol, from vanillyl alcohol and guaiacol. In this process, they also created an oligomeric coproduct. This coproduct they used to form a low molecular weight Novolec thermoset epoxy, which they then compared to the industry standard. The epoxy they formed could tolerate slightly lower temperatures than the industry standard, but could withstand higher pressures. They believe the product's stability is due to the aromaticity and the hydroxyl groups being predominantly para-directing. The research could lead to renewable and less toxic BPA and epoxy alternatives.
Interest in atypical bonding situations has led researchers to target complexes in which a metallobasic gold center is hydrogen bonded to a nearby OH functional group. Previously, hydrogen bonds were thought to be restricted exclusively to nitrogen, oxygen, and fluorine, but recent studies show otherwise. Through a series of gold derivatives, researchers have confirmed the existence of the bond between gold and hydrogen. The techniques that researchers Yongjin and Gabay use to confirm the bond's existence include X-ray diffraction, HNMR spectroscopy, IR spectroscopy, and extensive computational modeling. In addition to identifying the atypical bonding situation, these results also indicate that increasing the acidity of the OH functional group does not lend stability to the gold-hydrogen interaction.
Despite the WHO's estimation that more than 3 billion people are at risk of acquiring malaria and more than 200 million acquired the infection with Plasmodium falciparum in 2019, the current method of detection requires highly trained professionals undergoing laborious microscopic analyses that vary in detectability from less than 10 to 500 parasites per microliter of blood. Sanchez-Cano and colleagues report a new malaria detection method that lyses the blood cells to release Plasmodium falciparum lactate dehydrogenase that can then be detected through magnetic chemiluminescence. They begin with a 3-step detection method and move to optimize it for time. Here we see the shift from a 3-step detection to a 2-step optimization. This proved that signal and sensitivity were preserved. The next 3 pictures are the many steps that they took to ensure that they were reaching the highest levels of detection possible, ultimately achieving a 15-minute test with limits of quantification at 0.58 nanograms per milliliter of blood. The last picture shows their first clinical use of the test, in which it proved its reliability at the point-of-care interface.
Aging is a natural biological process with a 100% mortality rate. Much research has been done on the development of anti-aging drugs to prolong human life. However, there is a shortage of effective tools to test the effectiveness of such drugs. Jihon Lee and his associates have developed a highly sensitive fluorescent probe that can test an anti-aging drug's effectiveness by detecting the presence of beta-galactosidase, which is a biomarker for the aging process. When the probe detects the presence of beta-galactosidase, it emits fluorescence that can be seen using fluorescent imaging. Studies were done on both the cellular and organ levels using 3 groups of mice, a control group, an aging group, and a treatment group. The study showed that the probe was effective in detecting beta-galactosidase, as there was a bright fluorescence emitted by the aging group, but not by the control group.
Pigeon squabs, which are pigeons up to 4 weeks old, are an important domestic bird for the meat industry. Due to the lack of knowledge of the composition of pigeon kraut milk, the pigeon industry has been unable to achieve intensive breeding. Kraut milk is an important source of nutrients for squabs, especially in the first 7 days after hatching. This study was able to break down the surface level composition of pigeon kraut milk, which showed a 50% protein content compared to mammalian milk, which had 30% protein content. 2,558 proteins alone were identified in the crop milk among the 3 days that samples were taken. Levels of compounds in the crop milk were relatively the same in days 1 and 3, with a dramatic decrease in day 7, showing that the first 7 days have the highest nutrients content and are also the most important for young squabs. The researchers were also able to plot out the interactions of the proteins in the pigeon crop milk, which forms a spiderweb-like graph as shown. Further research would need to be done to identify specific compounds in pigeon crop milk.
Western Siberia has a very large oil reserve with the potential 4 trillion barrels of oil. To put that into perspective, the U.S. uses around 25 million barrels a day, which would be enough oil for about 465 years at current usage. Elena Mukina and co-workers, a Russian research group, used numerical simulations for an enhanced oil recovery method selection for shale oil. There were 3 methods that were applied and analyzed. Of the 3 methods, they discovered that huff and puff, where pressurized, heated water was periodically injected with a subsequent withdrawal of oil, was the most efficient extraction method. It used a surfactant solution which can reduce the interfacial tension so it decreases capillary pressure and then allows water to remove the trapped oil as a result of water bypass. As a result, it led to an increased cumulative oil recovery of 6%. With the simulation, they did a single horizontal well, which underwent a 9-stage hydraulic fracturing over an 8-year period. Hydraulic fracturing today is commonly used around the world. The way fracking works is they drill vertically to a certain depth and then drill horizontally. For the simulation, they drilled anywhere from half a mile up to 2.5 miles horizontally. As a result of the numerical simulations, they predicted a 3-fold increase over a 43-year period of time.
Previous studies have shown that kappa opioid receptor agonists relieve symptoms of multiple sclerosis by promoting oligodendrocyte differentiation and remyelination and immune cell modulation. The goal of this study was to find different agonists with better side effect profiles. So they looked at plant-derived cyclotides, which contain dulciphide-rich cyclic cysteine knots that are stable against thermal, proteolytic, and chemical degradation. It was found that of these, CAR-10 bound best and inhibited cyclic AMP production, making it an effective kappa opioid receptor modulator. The same procedure was used on T20K, which is a clinical candidate for treatment of MS, and it also bound to kappa opioid receptor and inhibited cyclic AMP as a full agonist. This shows that the plant-based cyclotides, with a better side effect profile than current medications, can be utilized as kappa opioid receptor modulators for the treatment of pain and axonal degeneration.
MALDI-MS is a common technique used to detect proteins in situ less than 80,000 kilodaltons in size. The common technique uses a small variety of matrices that are essential for the prevention of degradation of the analyte by the laser. The limits of these matrices are what puts the cap at the 80,000 kilodalton range. With the use of caffeic acid as a new matrix presented in this article, that detection range has been expanded to 200,000 kilodaltons. The caffeic acid matrix has properties that include strong ultraviolet absorption, micrometer-sized matrix crystals, uniform matrix deposition, and high ionization efficiency of the proteins. This new matrix allows for observation of proteins never before seen using MALDI-MS. Here it was used to observe the concentrations of high molecular weight proteins in the corresponding parts of a rat brain, Capparis masaki seed, and a germinating soybean seed.
Candida albicans is a major cause of systemic infections in immune-compromised patients. It is responsible for about 30% of severe infections, with a mortality rate higher than 40%. There are currently only a handful of antifungal drugs available for use. All have side effects and are toxic, and multi-drug resistance has rapidly developed against them. In this paper, it was found that when rapamycin, an antifungal that is mainly used as an immune suppressor, is mixed with peptide-like compounds, it has hypersynergenic antifungal activity, and that it worked against both drug-sensitive and resistant C. albicans isolates. This mixture can achieve 90% growth inhibition of C. albicans with only 0.002 micrograms per milliliter of rapamycin. The peptide-like compounds tested in this paper are all non-toxic, so they are suitable for clinical use. This means only a non-toxic peptide drug additive is needed to lower the dosage, while also making rapamycin more effective.
Chemotherapy is the most commonly used treatment for lung cancer, but chemotherapy resistance is one of the main causes of treatment failure, and a combination regimen may be an effective way to overcome this. Most patients develop resistance after several months of treatment with cisplatin, referred to as UDP, leading to treatment failure. During this work, an Aspergillus nigrofungal strain was isolated from the gut of the woodlice species Armadillium vergeri, otherwise known as the roly-poly. From this strain, 20 polyketides were isolated and compound 12 was found to be the most useful. The chemotherapeutic resistant cell line is the A5490DP cell line, and the results of synergistic effects were shown in Figure 6. Resistant cells exhibited high cell viability after treatment with DDP or 12 alone, but cell viability decreased significantly after combined administration. Chemotherapy resistance can be caused by multiple factors, including elevated glutathione or GSH levels. GSH has a detoxification effect and simultaneously converts to oxidized glutathione, known as GSSG, which regulates the intracellular redox homeostasis. The results of a GSH-GSSG ratio analysis in Figure 7 showed that compound 12 reduces the ratio of GSH-GSSG, destroying the redox balance of A5490DP and promoting cell apoptosis.
A new glucose monitor was made non-invasive by testing interstitial fluid. Ionic compounds were introduced to the body through the skin. Interstitial fluid extraction was used to detect glucose levels in a non-invasive way. In other words, no finger pricking. The new glucose patch is worn like a temporary tattoo, made of electrodes covered in hydrogel layers. The solid aerogel circular-shaped gel was formed and stuck to the skin. The sensor was powered by iontophoretic and electrochemical subsystems and shared a common connection to the sensor. Even though technology is very advanced, the IR sensing tattoos is only a single measurement operation. It can only detect 4 hours of blood glucose levels and interstitial fluid glucose levels of diabetic subjects. However, the patch can be used for up to 8 hours on non-diabetic patients that undergo the same tests. Non-invasive advances like this help patients become more willing to manage diabetes and improve their quality of life.
Gram-negative bacteria represent a significant danger as opportunistic pathogens in hospital settings, and antimicrobial peptides can assist in the fight against them. In this article, the authors identified and described 2 new antimicrobial proteins within the genome of the ghost pepper. They used a powerful combination of in silico statistical modeling and peptidomics to identify potential peptides within the genome, then isolated and characterized the peptides, including their sequences, shown above, their mechanism of action, non-existent toxicity to human cells, and 50% inhibitory concentration against several gram-negative bacteria, also shown above. The minimum bactericidal concentration of the peptides against A. baumannii was found to be 8.6 micromolar. In addition to the information gained about 2 specific peptides, their results demonstrate the exploratory power of the combination of statistical prediction and peptidomics.
Cervical cancer is a prevalent disease that infects many women. More frequent and better testing methods are needed to propel female reproductive health to a new level. The current detection methods are invasive and can cause pain. These include procedures such as a pap smear. Matali Bosk and colleagues have determined a new, non-invasive method to detect abnormal cells. The new method involves collecting a urine sample and nothing more. Gold nanoparticles are bound to an antibody which will bind to an antigen, or in this case is the abnormal cervical cells. The antigen is detected using a portable instrument that uses visible light. This new method allows for more rapid detection at lower levels and also will be available for use in the clinical and all point-of-care contact settings.